cut this a little short. All right, so we were looking at flower anatomy. And remember, don't be surprised if you were to see like a diagram of this on the final exam and ask you to, label. So please start learning the different parts if you have not ever done so. And then the other thing we looked at too, I said, make sure that you're comfortable with this idea because this follows the trend of evolutionary process of flower design, how we see ancient flowers had more superior ovaries. And then as the evolution of angiosperms kept going, we see a shift to inferior with the more advanced flower types. And we'll look at some ancient flowers and more advanced flowers in a little bit. Okay, so let's keep zooming into the ovary. And so if you look at the female part of the flower, they're showing over on the far left a drawing of the stigma, style, and then an ovary. And then if you take that ovary in cross section, that's what you see here, this image, and then also the microscope image on the far right. So that's a cross section of an ovary. And in this case, you can see there's 1, 2, 3, 4, 5, 6 ovules in this ovary, okay? And so they have it labeled over here on the left for you to look at. So the ovules, we have to think about what's happening in there because that's pretty precious area. That's where we're going to get the egg to develop eventually. So it's in these ovaries. So again, be comfortable with identifying. If you see a picture like this on the final exam, whether it be the lab lecture or lab exam or lecture exam, oh yeah, these are ovules within an ovary. Okay, so start training your eye to that. So what we're going to do, we're essentially zooming in now and looking at how is the megagametophyte going to develop in these ovules? How's that going to happen? So what you're seeing here is pretty early stages, not a whole lot going on here. So we need to think about how does the megagametophyte or the female gametophyte develop? Now, in the angiosperm realm, many plant biologists use the term embryo sac. Okay, so embryo sac is the same thing as a megagametophyte. Same thing. It's just for some reason they switch. Up until this point, we've been talking about gametophytes and megagametophytes, and now they have to switch out on us. And so you're going to see a textbook probably prefers embryo sac as well. I tend to prefer megagametophyte because we've been doing that up to this point. We might as well just stay consistent. So here we're looking at the idea of we still have to think about pharangium tissue. We still have to think about spores inside the pharangium that eventually go through meiosis. Now, with the gymnosperms, we learned that all those spores reduce down to 1 in the female. There's just 1 megaspore mother cell. We see that again with the angiosperms. 1 megaspore mother cell. So that's what they have here labeled in the center of this very young ovule. And so that megaspore mother cell, there's just 1, just like in the gymnosperm. That cell, that megaspore mother cell has to go through meiosis and produce 4 cells. When you have 1 cell going through meiosis, you end up with 4. And again, you're going to have, well, actually, okay, let me work through this. I'm going to show you a close-up. Here's the megaspore mother cell. And let me show you pictures as I talk through it. Okay, so here's the megaspore mother cell. Here is the young ovule. So here you have the megasporangium tissue. And in this case, it's just going to be 1 megaspore mother cell. And so right now what they're showing is the result of that megaspore mother cell going through meiosis. So when that 1 megaspore mother cell goes through meiosis, you have 4. And you can count those up. There's 4. Only 1's going to make it again. So 3 die off. They're trying to show that 3 are dying off. And you're left with that functional megaspore. Okay. And then you still have the same idea like what we talked about with gymnosperms. We have that integument tissue surrounding all of that to protect it with a little tiny opening, the micropod. So some of the same ideas here is what we just talked about in the gymnosperms. Okay, so now that functional megaspore, what is it going to do? It's a little different now when you compare it to the gymnosperms, because in the gymnosperms, there was a lot of cell division going on. Lots and lots of cell division. They even have a stage of all that called the free nuclear stage. You just lots and lots and lots of cells developing. We don't see that with the angiosperms. They simplify things. Okay, so you see this functional megaspore. All it's going to do is go through 3 rounds of mitosis. That's it. So if you have that one cell, that functional megaspore going through 3 rounds of mitosis, what you end up happening, you end up with 8 nuclei from that process. And theoretically, there should be 8 cells that will form around each of the nuclei. But we don't see that. They got to be even more simple. So what they do is, by the time it's all done, you end up with only 7 cells, but 8 nuclei. So what that means is one of the cells actually gets 2 nuclei stuffed into it as it's developing. Weird, but it happens. All right, so what I am describing, and I'd like you to know that term up there, polygonum. What I am describing is the polygonum type megagametophyte or embryo sac development. It is the most common. So I said there's over 250,000 angiosperm species. This is the most common type, and this is the only one I expect you to know. So be comfortable with what's on this slide, walking through what happens to that functional megaspore. You end up with 8 nuclei. 2 nuclei get stuffed into 1 cell. So that means you end up with 8 nuclei, 7 cells. And then this very final picture here, I'd like you to be comfortable again with the details of all those different cells. So what they see when they look at this polygonum, when it's all done and ready to go, you're going to see that they migrate, those cells migrate to different portions of the megagametophyte. And so there's going to be 3 cells that go to the far end, away from the micropyle. And those are called the antipodal. I always think of it as they are the opposite or the anti of the micropyle. All right, so the antipodal cells, there's 3 that are called antipodal cells. And then where you see the micropyle at the other end, what you have are 2 cells that they've named synergid. Don't know why. And then the synergid has the egg right in the middle. And you can see that egg is like right up against the micropyle region. So it's much easier for the sperm to find that egg. As long as you get the sperm reaching the micropyle, it's pretty easy then to fertilize that egg. And then in the middle, you see those so-called polar nuclei. Those are the 2 cells that or 2 nuclei that get stuffed into 1 cell. And we call them polar nuclei. And they're going to be important. Okay. So please be comfortable with the anatomy, especially of the final picture here. I don't care that you know the phrase egg apparatus that's on there. You don't need to know that. And then what's the brown stuff surrounding it? That's the integument tissue. Okay, that's the integument. Okay, so this is the megagametophyte. You see that I did not say anything about an archegonia with an egg inside. Okay, there's really not an archegonia in the angiosperms. So that's different. All right, so now in lab today, you're going to look at some prepared slides of the lily species. Lilium genus, and that's the lily plant. And so you're going to see slides like this. Now, and they'll be labeled, but what you're looking at here is an ovule. And now you can see that there's some cells in here, and you're going to see that the slides have labels. Like this one was labeled 8 nucleate megagametophyte. So it's close to being mature, but it's really hard to tell with live, real samples like this. It's much easier in a drawing to get it. But when you look at actual samples, so it's not like we're going to fill this up and say, what stage is this in? We're not going to do that because it's hard. It's hard to tell. But you can see here, the reason they use lily is that the cells in there are pretty big. They're easy to see, but again, it's hard to tell, well, what phase is this really at? Okay, but we will have it, Emily and Olivia will tell you what order to work through these. So here's a close-up of that previous one. So the cells are nice and big. The ovaries are nice and big. The ovules are nice and big. That's why we tend to use the lily plant for this. But unfortunately, lily kind of has an odd little twist to they don't strictly do polygonum. So I'll just mention that in a minute here, what they do. So again, this is a real sample, but again, you can see it's kind of hard to, figure out what's what. And so I tend not to use real samples if I really want you to label something on an exam. It's too tricky. So it'll be more of an idealized drawing that you'll be working with. But they do a pretty good job here trying to outline where everything is. So this is like ready to go. So it's ready, the egg is there. It's ready for fertilization. Here's a more stylized drawing of it. It's a little bit more easy to figure out what's what. So now the job is for the male part of the flower that's producing pollen. We got to get that pollen delivered to the female part of the flower. And then we have to somehow get the sperm delivered down to the micropyle. And so it has to, the pollen tends to land on the stigma, the top part of the female portion of the flower. It'll land on the stigma, and then you have to have a pollen tube germinating and growing all the way through the style, get into the ovary, get into the ovules, and then find the micropyle. And then it can deposit its sperm. No water necessary, just like in the gymnosperm. No free water is necessary. This is an awesome picture. Again, to the untrained eyes, I wouldn't put this up on an exam and ask you what's going on. But in the notes, you can see they've captured double fertilization going on. So this little granular looking thing, that's one sperm that is trying to fertilize the egg. And then here in the central part, another sperm has come along and it's going to fuse with the polar nuclei cell. And so very interesting prep. Whoever got this done at their job should get a bonus because that's really, really hard to capture. So if everything goes okay, if that egg gets fertilized by the sperm, you're going to start to see an embryo. So that's what you see here. That little blob is the fertilized egg starting to go through division. Mitosis, mitosis, mitosis. It's got to grow into the baby. It's got to grow into the embryo. Okay. And so here is just the close-up. You can see that the micropyle area is sealed up, further protecting what's going on. Very precious stage of their life. So they try to protect it physically. Okay. So what we are describing here is the polygonum type, which is in the top row in this table. So that's the one I want you to know. So that's what we just walked through. About 70% of flowers, species, and plants will do this. The others are being shown here. Allium, you see lilium on here. Okay, so they're just saying, here are some examples of what other plants do, the other 30%. You're not going to need to know any of those. I will briefly mention what's going on with lily in a different slide in a little bit here. So what we're going to find out is that when you have this double fertilization going on, so remember now, gymnosperms, they deposit 2 eggs, only sperm. They deposit 2 sperm, only one makes it, fertilize the egg, the other one dies off. So those 2 sperm kind of race for the flag and go, and one makes it to the egg, the other dies. Angiosperm, different. Both sperm make it and have important jobs. So one sperm will fertilize the egg, which is over here by the micropyle area. Now this, what's going on in the middle here, that second sperm merging with the polar nuclei in that cell, what you have happening there now, what's going to result from that is the endosperm tissue from that. So that second sperm that's fusing with the polar nuclei cell, you have then that fertilized, or can't sound like fertilized, because you tend to say that term more with the egg, but that fusion event, say it's a fusion event of the sperm holding the egg, then that will go through mitosis, 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 and grow into the endosperm. Very, very important, because that baby endosperm for nutrition. That's what it's relying on. It's its bank of nutrients that it has to tap into when it starts to germinate. And so that embryo that's going to result from the fertilized egg really does need that second sperm to be successful, because you need that endosperm to develop so it can feed the baby. Okay. So I said that, excuse me, Lilium is a little different. A couple of things, and they'll go through this in lab a little bit too. And it's just tradition that Lilium are the prepared slides that you look at, because everything's nice and big. It's easy to see what's going on in terms of, it's just nice and big, easy to get into focus. Lily has to be a little different. So what happens is that megaspore mother cell is shown in A. Okay, that's the megaspore mother cell going through meiosis, and you see 4 cells. Now with polygonum, I told you 3 die off, 1 remains. Lilium's got to be different. All of what happens is 3 of those cells will fuse. It's really weird. 3 of those cells fuse, so then it becomes 1 big cell with 3 nuclei in it. Really strange. And then you have that 4th cell off by itself. And then these next stages, E, F, and G, that's the 3 rounds of mitosis. So they still do 3 rounds of mitosis, just like polygonum, but you have that fusion event first, where all the 4 cells participate. You don't have 3 dying. So a little weird. But the end result of that is it makes whopping big cells easy to see under the microscope. So I think that's mainly why traditionally we use the lily species. And you can still see they're migrating similarly to what we saw with the polygonum type. When everything's all said and done, you still have that. It's just that these antipodal cells will be, they call it triploid, which means 3 nuclei. And then you can see the 2 in the middle, those are your polar nuclei, and you can see one of the cells is triploid, the other cell is just 1 end, it says 1. And so what happens is you have 1, 2, 3, 4 nuclei in that center cell, the central cell. And then you have to imagine the sperm coming along with its nucleus. So that brings it up to 5. So what ends up happening is when that sperm makes it there, you're going to end up with 5 nuclei. And so they denote that as 5 times n. And they call it a pentaploid condition. You know, so far we've been talking about haploid, diploid. Now we have to get into triploid and, in this case, pentaploid. So that means the endosperm tissue, each cell in there is actually pentaploid when you look at lilies. Okay, so it's a little weird. Okay, let's think about the male part of the flower, the microgametophyte. What's going on there? So we need to find the anthers. So you tend to see anthers, when you look at them more carefully, you'll see that there's usually 4 microsporangium chambers or distinct regions, 4 microsporangium clustered there into 1 anther. That tends to be the pattern. So look for that pattern of 4. And then remember, the filament is like the stalk that will hold the anther up high enough, hopefully, for pollinators to come along, maybe wind if necessary. All right, so this is all microsporangium tissue. Now, before it becomes pollen, we have to back up a little bit. So we say this is microsporangium tissue, and we have microspores. So this, you have to imagine, first there's microspores, eventually pollen, but first it's microspores. So let's look at this actual slide, a cross-section of the flower. And you don't see the full thing because it was too big. But this is the center of the flower right here. This is the female part. Okay, this is the female part. And so this is the ovary tissue, and it has 1, 2, 3, I think 6. It's very young here, 6 ovules. And then here you can see 4 regions here. And if you can train your eye, that if you see that pattern of 4 like that, that's the anther that you're looking at. Okay. So we're zooming into what's happening in each of these microsporangium regions. So we have to think about the microspores. So the anthers, what you have is you have a microspore, many microspore mother cells. They don't get reduced down to one. There's lots. So you have lots of microspore mother cells. Each one will go through meiosis and give you microspores in the haploid state. Then each one of those develops into pollen. So that's why you get lots of pollen, because you end up with using lots and lots of microspore mother cells, all going through meiosis, all producing 4 microspores. And then what you see happening here is that each microspore will develop into the mature, or well, into a pollen grain. Let's just say that. So right here, what you're looking at, where my laser pointer is, that is a young pollen grain. And you can see there's only 2 cells in that pollen grain that have developed to this stage, the generative cell and the tube cell. That's different from the gymnosperms. We said gymnosperms have 4 cells that make up a pollen grain. Now the angiosperm says, let's simplify it. Let's just do 2. All right, so they reduced it down to 2. So this pollen grain is immature because you don't see any sperm yet. Okay, nowhere did the artist put the word sperm in there yet. So when pollination happens, whether it be a pollinator, a bee that came along and got some pollen on its body, or wind, it's immature at this stage. So pollination will be immature pollen grains getting transferred, and then eventually you'll start to see that generative cell will divide and produce 2 sperm. Okay. So pollen grains get released or get transferred by a pollinator in the immature state. Hopefully, when you look at the angiosperm definition of pollination, it's getting transfer of the pollen to the stigma of the flower. So let's look inside the anther again, more advanced. So now you see a lot of, these are actually pollen that are ready to go. They're ready to be transferred to the stigma. They're still immature. There's only 2 cells, no sperm yet, but that's okay. Eventually they'll get the sperm going. So you have to imagine maybe a bee comes along, brushes up against the pollen, and then it crawls around on the flower, and the pollen gets brushed up against the stigma. Then what happens is that generative cell will divide and produce 2 sperm. So that's what they're trying to show here, the generative cell finally dividing. And then the tube cell's job is to grow out into a tube to deliver the sperm. So once it lands on the stigma, that will kick in if everything's going okay. You'll get, there's some chemical signaling usually that goes on between the stigma and the pollen. And if everything's a green light, then the tube cell starts to grow down the female part of the flower. It's got to reach the ovary and the ovules where the megagametophyte is. So pollen grains can be very, on the surface, very diverse in how they look. They have a very tough coating of proteins that make pollen grains capable of being, you can revive them thousands of years later. So they're very, very tough and able to withstand years before they can get, if you try to actually pollinate with them after thousands of years, they still can do it. The pollen grains are so unique for each plant species that they've used them in for police work, looking at solving murders, where they found pollen on different suspects and could trace it to a certain region of that area where only this type of plant grows, and they were able to convict murderers based on pollen grains. That's how unique they are, and it held up in court. So they had a forensic plant biologist actually take the stand. That case study that I read, a young man murdered a woman, and he transported her in the bed of his truck, and the pollen grains came with. And so they were able to find the pollen grains in the truck, even though he burned his clothes. And so they were able to map it out and trace it, and he got convicted. So one really good use of pollen grains from our perspective is that they can, they're so unique that you can use them in that manner. Here's a pollen grain with one opening. The fancy word for that, do they have it on the previous slide? Aperture. Aperture is the fancy word. The aperture is basically a hole or opening that's going to allow the tube cell to come out with the sperm. And it's very pretty characteristic. If it's one aperture, it's probably a monocot plant that this came from. A monocot has one aperture. A dicot, this is a dicot pollen grain, and you can see 3 here. There's one, here's another one, and then the third one's kind of tucked there. You can see part of it. So those are 3 apertures. If pollen grains have 3 apertures, it's a dicot. That's a pretty good rule of thumb. So please be aware of that. And you can see the difference in the surface morphology. It can be pretty simple, like what we saw with the monocot. Here you have some interesting morphologies. Here's some other examples. So quite beautiful when you look at the different types of pollen grains that get produced. Here's some more. So here you can see the monocot on the bottom right. Can't tell what the bottom left is. I can't see the apertures. Does anyone, is that an aperture maybe where my laser pointer is? To the right of that, does that look like an aperture? Yeah, it looks like a black hole there. I think that's a monocot, but it's hard to tell. I don't know what species it is. And then the top 2 are dicots. So very, very beautiful, intricate designs, or like this bottom right, pretty simple. It runs the full spectrum. But the job of the pollen grain is to find the stigma. So what is being shown here is pollination. So transfer of pollen from the anther to the stigma, that's the definition of pollination in the angiosperm world. Okay. Transfer of pollen from the anther to the stigma. And you can see lots of pollen grains can land there, and they can all start germinating out their tube cell. And you can see the one in the middle here seems to have gotten a head start. And so that tube cell is going all the way down the style. Got to make it all the way down there, all the way into the ovary, find an ovule, find the micropyle. And by then the megagametophyte inside there is ready to go, the egg. And then you'll get the 2 sperm discharged. And both sperm are important. Both have a really good, important role. One fertilizes the egg, the other one will fuse with the polar nuclei. And from that polar nuclear fusion event, you get the endosperm. So here is the complete life cycle. I believe this one's from your book. They're showing both the female and the male development from, first you have the megasporangium tissue and the microsporangium tissue. And then after meiosis, you start to see the further development like we just talked about. So in lab today, you'll work through these. If you can have this life cycle up on your laptop, and I think your lab manual has one as well, but it'd be a good idea to have it available as you're looking through the slides, so you can kind of step by step work through this process from start to finish. Questions on the angiosperm? Yes. So is this the polygone? Yes, this is showing the polygone. Yes, this is the only one you got to know. And in lecture. In lab, they might ask you on the final exam about the lily with the fusion event. I don't know, ask them if they're going to do that. It depends on how they emphasize it. They have a little leeway on that. So as you're studying this, of course, on the final exam, you're going to have seedless vascular plants on there. and then the gymnosperms and the angiosperms. So be a good idea to maybe, make sure you're doing a little bit of compare and contrast, like what's similar of how they do this stuff. And like when you look at the gymnosperms and angiosperms, there's some similarities, but there's definitely some differences. And so I think studying that way, as you're looking at it and trying to compare and contrast, it does give you, I think it sets the information a little firmer in your head and helps you to get a little deeper into the content for better learning. And so I do highly recommend that, because I'll be asking, like, when the pollen is shed from, in the male cones and the gymnosperms, how many cells are there? 4. Well, what about the ants? Well, 2. So these are differences that make it's a big deal. I know it may seem weird to you, like, why is that a big deal? But in the evolutionary process, these shifts like this, these are like landmark differences, and that can point to why the angiosperms are so successful. They're the most dominant species or group, phylum, plants on the planet. And so we're interested in tracking what's similar and different, and those differences that we see with the angiosperms, all of those added up really help them to be the most successful in terms of the sheer number on Earth. Okay. So this is the polygonum type. But again, it's a lot of the same terminology that you have been seeing, except here with the angiosperms, we really don't talk about archegonium. We don't see that really happening. And so that has gone away. And of course, flowers, huge innovation to have flowers. This next 2 slides are just the previous slide cut up a little bit so you can enlarge that when you want to. This is not from your book, it's from a previous edition, I believe. But again, for study purposes, it has a lot of words on there showing what's happening. It's the same basic idea of what we just looked at. If this works better for your brain as you're learning it, great. Either one is fine. It all has the same information. Oh, I forgot to introduce that term. So I talked about it. I said how one of the sperm fertilizes the egg, the other sperm fuses with the polar nuclei. The fancy term for that is double fertilization, which is a little weird because polar nuclei really don't fertilize. Weird, but that's the phrase they came up with. So double fertilization just means that both sperms that get delivered by that pollen grain, both sperm participate in very important jobs. Okay. And so one of the fused sperms with the polar nuclei develop into the endosperm, and then that second sperm that fertilizes the egg, then you get your zygote, and then your embryo, and then the sporophyte eventually. Okay, so again, the sporophyte dominates in the angiosperm. That's what you see. Just like with the gymnosperm, the sporophyte dominates. You're out hiking around and see conifer trees, that's the sporophyte. And then the gametophyte is maintained on the plant. It is here too. It's maintained in the flower. And those spores now don't get released. It's all retained and protected. Okay. This last, this image too, again, it's not from your book. I just put it in for study purposes. Same basic idea. Okay. Here you're looking at very simple drawing here where you have the fertilized egg. So they're calling it the zygote. So if it's a zygote, that means the sperm made it and successfully fertilized the egg. And then you can see here in the middle, the second sperm fused with those 2 polar nuclei. So that's triploid. In the polygonum type, it's triploid, 3N. So the endosperm is all 3N tissue. If you look at the endosperm cells, every single one of them has 3 nuclei, which is weird, but that's the way they do it. All right. And then what you have to imagine now is, so that's a success. Both sperm played a role. They did their job. And now that those 2 polar nuclei and the sperm, all of that's going to develop into the endosperm, which you see here over here on the right is dark green. And then that fertilized egg develops into the embryo or the baby sporophyte. The integument starts to shift as well. It has to toughen up and become the seed coat. So that integument tissue will develop into the seed coat, which is very important. That's a very, very important part of the seed. And so all of that has to go correctly as well. And so the endosperm provides nutrition. Back when we looked at the gymnosperms, it's actually the megagametophyte in the gymnosperms that's providing nutrition. Okay, so that's a huge difference. And then also when you look at the megagametophyte development in the angiosperms, really straightforward. You get 3 rounds of mitosis, you're done. With the gymnosperms, they go through that huge free nuclear stage and it's pretty complicated. So the angiosperms kind of simplified everything down. So let's make it simple. Just go with 3 rounds. And you've already studied seed. You'll look at them again a little bit today in lab as well, just to refresh your memory. Thinking about the anatomy of the seed, you need to think about the embryo, of course, the baby sporophyte inside. And then depending on the seed, you might have really obvious cotyledons. You might have more slender cotyledons. You might have endosperm tissue. Sometimes though, the endosperm tissue kind of gets subsumed or taken over by the cotyledons. Like kidney bean, when you crack open a kidney bean seed and look inside, there's really no endosperm, it went away. The cotyledons really took that away. And so the endosperm doesn't always persist, but if it doesn't, you'll definitely have some nice cotyledons taking its place. And then sometimes you have both endosperm and cotyledons. So it just depends on what plant you're looking at. Okay, so you'll look at some of those again in lab. And of course, you know, the goal here is, and we'll talk more about this next week, we'll get into pollination strategies, seed dispersing strategies, but that plant, from the plant's perspective, wants to get that seed fully mature and ready to go, and then encased in fruit tissue. And then one way or another wants to get that seed out somewhere else, away from the mother plant, where hopefully the seed will then germinate and grow. And so here you're seeing different strategies of germination. Sometimes the cotyledons comes above ground and other times it won't. But again, it's just different strategies of how that will germinate and grow. I'm not going to ask you terms here. So the, other than like cotyledon, but, and you know, the basic anatomy of the seed I want you to know, like cotyledons, endosperm, seed coat. But these other terms, like over here, coryza, oh, the radical, knowing that the radical is the first thing out, that's the embryonic root. But I don't get into like hypocotyl, epicotyl, we're not getting into that. So some of this other anatomy terms, I'm not gonna ask you about those. So as you're studying, if you run into some terms that I really didn't specifically mention and you're wondering, just ask me and I can let you know if whether or not you need to know that. Okay. Questions on any of the seed anatomy or polygonal, embryo development, maybe the mesocyte? Then the exam Monday of final week. Yep, that's the last exam. Final exam Monday at noon. So just like our lectures at noon. Be Monday at noon in this room. And oh, I sent out about homework assignments. Okay, so make sure to pay attention to those deadlines. And then I'll have credit next week. I'll post that one. Okay, so please make sure to do that. And then I started with questions if you're unsure. I know this is it's a lot. This part of the semester, it's pretty stressful. So let me help you as much as I can and answer any questions. Okay. This is the end of the slide set. I guess we'll stop here. It's a convenient place to stop. And next Tuesday, we'll get into looking at flowers and then eventually we'll get into fruit. Right, so we'll start looking at that information. My goal, by the way, is to have like the last lecture period. I'm hoping I can hold a review session during the lecture period. That way you can all attend. So let's hope we can get that, swing that to happen. All right, have a good weekend.